Dolly the sheep was named after Dolly Parton's boobs, and she wasn't actually the first cloned animal. Way back in 1885, a German biologist Hans Drake made artificial twins of sea urchins. He waited until the urchin embryo had undergone its first cell division and then shook the cells apart. And the two cells subverted expectations by becoming two fully formed sea urchins. And then in 1902, another German, Hans Spemann, figured out how to make artificial twins out of salamanders, which have stickier embryos. So he took a strand of baby hair and made a little noose out of it. Then he tightened the baby hair noose around two cells of an embryo until they separated and became two identical salamanders. But both of those were artificial identical twins, genetically identical but made by separating early embryonic cells from each other. As a side note, when World War II came around, Drake was openly hostile to Nazism, but Spemann, when he accepted his Nobel Prize, gave the Nazis salute. Back to the early history of cloning, in 1958, British developmental biologist John Gurdon made clones of tadpoles. This time he used somatic cells, which are not stem cells or gamete cells from the intestines of a tadpole. In a process called nuclear transfer, he transferred the intestinal cell's nucleus into a cell that had had its nucleus removed. It was a big deal because it suggested that cells retain all of their genetic information even after they've differentiated themselves into specialized cells. And one of the first times clone was used in reference to an animal was when a colleague of his was describing the results. Okay, now we get to Dolly the sheep. She was fashioned by Keith Campbell and Ian Wilmot of the Rosalind Institute in Scotland in 1996. She was the first mammal created through the process of nuclear transfer with somatic cells, or adult differentiated cells. They took a cell from a sheep's udder and fused it to an egg cell which had had its nucleus removed with a small electric shock. It took 277 attempts, but eventually one of those cells was carried to term by a surrogate mother. And the resulting sheep was named Dolly, and she lived pretty healthily for a short 6 years. And to bring it back to the beginning, Ian Wilmot said, "Dolly is derived from a mammary gland cell, and we couldn't think of a more impressive pair of glands than Dolly Parton's."